Cool. Glad to see you all back. So before we get started, we can maybe take a couple of minutes to just see if anyone has any questions from last time. So feel free to just unmute yourself or type in the chat if you have some remaining questions from Monday.
Hey, Jordan.
Yeah.
The Word document you set out, it's got kind of, ideas for homework that was attached to each week, and then your
Yeah.
slides. Is it just changing? Is that kind of the idea?
Yeah, sorry, I haven't updated that since then. That was more of just a rough draft for the schedule. So I would just focus on the homeworks that I've put in the slides. Those are kind of the final drafts of the homework.
Cool.
Okay. Well, if there's no other questions, we can get going. Okay. So today we're going to talk more about some of the common file types that you'll see when you're dealing with just kind of your run-of-the-mill sequencing data and other kind of biologically relevant data types, and how to work with them, particularly in the command line context. So, like that, okay. And again, the slides from today, they are on GitHub, which is in the general Slack channel. So if you want to access those, they're there. I think the PDF and the PowerPoint for today is both up. And then it seems like the video upload for the recording process is pretty quick. So I think usually within an hour or two after class, that should be available online on that YouTube channel that's in the Slack channel. Okay. So I want to start off talking about our package managers. So when you're processing sequencing data, there's a lot of tools out there that have already been developed, that have been well tested and kind of vetted by the community to make sure that they're accurate. And so a lot of these tools, for I guess the developers, in an effort to make them more accessible to the community, they'll often offer them via this package manager format where they can be downloaded. And you can think of this kind of like the command line's app store. So say I want to use a sequencing read alignment program. One that we'll talk about later is called STAR. I can use this package manager called Anaconda or Conda, which will allow me to just quickly download that, and it kind of handles all of the intricacies of getting it set up in the command line so that you can basically, once it's downloaded, you can start using it out of the package. So what we're going to do is download Conda, and I think the URL is up there. I didn't copy it here, which I should have, but so when you're downloading files to your desktop or the supercomputer using terminal, most operating systems will come with something called curl. I don't remember what the C is, but then the URL part is for like a web URL. So basically this is going to allow you to fetch whatever file is at the end of this URL here. So I am going to quickly, oh, it's not happy. Let's see. So when I do this, there's a couple different common options to be used, but if I just want to output that file from the URL as is with the same name. I'm just going to use a dash capital O. And then I'm also going to use, so I could either give this as, I think I actually have to, so dash O L. So then this is going to also use this capital L argument. And basically what this is going to do is, just in case the actual file at the URL moves somewhere else, it will kind of trace the file down the rabbit hole of URLs until it finds the actual file and not just a link to where the file got moved. So then I'll enter this URL. I'm gonna have to hand type it since I don't have this quickly available. And then I will use the Linux distribution since the supercomputing environment is Linux based. If I was on a Mac, I would, instead of Linux here, I'd put Mac OS. And then I'm going to specify the build information .sh. So as you can see, when I run that, it'll kind of give me this download progress bar. And once it's downloaded, if I type ls to list all the files, I will see that file that I just downloaded here. So you notice that this is, the file is ending in a .sh. So this stands for shell script. So we talked about last time, anything followed by these or anything after the $ is a command within terminal. And so the way you execute one of these scripts is you just type bash and then mini conda, and I'll just autocomplete. And so bash is the command that says, I'm going to give you a shell script, run the shell script. So I'm going to hit enter. And so this is kind of nice where it kind of walks you through the installation steps. So just says, okay, we're going to install conda for you, press enter, I press enter. And then you have to scroll through. I can just scroll with my mouse, it looks like. Maybe enter. So I'm going to scroll to the end of the license. Okay. It'll ask me, do I accept the license terms? Yes, no. I'll type yes. And then it's going to ask me, are you sure you want to download this to this location? And so this is the pathway to my my basically home directory within the supercomputer. And then it's going to make a new folder called mini conda 3 where it's going to store all of the conda packages that it downloads. So I'll hit enter. And because it already exists, it's going to stop there, but you just basically press enter the whole way through or yes, and then you'll be good to go. So now if I type conda, and then unlike a native command line program like ls or cd or the make directory mkdir, when you're using a kind of external tool like conda or some of the other sequencing tools we'll use, usually you'll have to type -- help to access the documentation for this tool. So if I run this, it'll give me some output. And so this is saying that conda was installed, it knows where to find it, and it's operating fine. If for some reason there was an issue with the installation, this wouldn't have showed up. I would have had to troubleshoot through that. Okay, so now conda is set up. I can download all the packages I would want for analyzing some sequencing data. So we'll go here. So another advantage of using these package managers is you can develop these different environments, computing environments. So basically any package that you would install. Okay, so someone asked a question, they got a message if the installer should initialize miniconda by running conda init. Yeah, you should answer yes to that. So that's basically just going to do some behind the scenes processing to make sure that conda can be accessed no matter where you are on the supercomputer. Okay, so like I was saying, an advantage of conda is we can, I guess, set aside these different computing environments or locations. So maybe I'm using a set of tools for some kind of sequencing analysis. And then I have another set of tools that I only use for something else. And maybe one package isn't compatible with another package, so I can't install all of those packages in the same location. So I can kind of partition where I install those apps by creating a conda environment. So here I'm going to do conda create, I'm going to say name, and so I'm just going to make an environment called class. You can name this whatever you want. Like if I'm doing maybe RNA-seq, I would just call this RNA-seq. If I wanted one for proteomics analysis, maybe I just call it proteomics. And it's important that in these names, you don't have any spaces in the names. Just put like an underscore or something there. So I run this. So I already had one called this. I'm just going to overwrite it. And so it's going to initialize this environment with some of the basic packages that it's going to need. So then it finishes setting up that environment. I'm going to proceed. So then it will install the packages it needs just to that one environment called class. And now anytime I want to use this environment, I can just type conda activate class, and that will activate the conda environment. You'll see here before I ran that, it had this base here. So that was saying you're in the base environment. Now I'm in the class environment and it shows that there. Something important to note is if you were to log out of the supercomputer or open a new window and you wanted to use this environment, you need to run conda activate class again to reload that environment. Okay, so I have my environment. I've activated it so I can start using it. And now we can start installing packages. But just one more caveat is that there are, so basically within the package manager, there's different channels for different categories of programs. So you'll see here, BioConda is for bioinformatics packages. Conda Forge is for, I guess, more science engineering, but not biology packages. And so basically I want to set these channels by default so that anytime I start installing something, it will just search through these channels to find the appropriate packages. And I need to do this for every environment that I have. So if I want it to be able to search through these, then we'll have to set this. So I want it to set the channel priority to true. So I'm going to run conda config set channel priority true. And then conda config channels bioconda, and then add conda forge. Okay, and again, this is already on the list since I've already set this up. So now I can go ahead and install SAM tool. So we'll talk a little more about this package later. But so I run this command and it will, let's see. Okay. So essentially what it's going to do is search for the package, search for all dependencies that this package relies on. So any other packages or libraries that it needs to be able to run. And then it'll give you the summary of what's going to be downloaded, what's going to be updated or installed in order to make sure that the program you're downloading will function properly. So I'll say yes. It's going to download, extract, and install all of the packages. And then, so now if I were to run samtools -- help, now you can see that samtools is installed in the environment and I can start using it down the road. So this is just a screenshot of what we went over. Okay, and we talked about, just to kind of reiterate, instead of using man and then the command, we're going to be using the command name followed by help. And so you'll also see that within samtools, there's a bunch of sub packages essentially. So if I wanted to find out how to use samtools view, I could do samtools view -- help. And then it prints off the different arguments and options that I can use with samtools view. And we'll talk about this a little bit later, actually. Okay. So that's how you'll work with a package manager. Are there any questions about what's going on here or anything like that? Okay. So let's talk about a couple more command line commands that will come in handy on the supercomputer, whatever, to basically move your data around and set it up for actual processing. So I'm going to go back into that test folder that I made last time. Let's just see what's there. And so say I wanted to move this file somewhere else. Can't remember if we talked about this last time, but we'll just kind of reiterate. So this can be used either to move a file, rename a file, or do both at the same time. And so say I wanted to move this file to this away folder. So I could say move file space away. And now we can see that file is no longer there and it's in the away folder. Okay. So there's a conda question. We'll come back to this at the end. Remind me to cover the errors with conda after we talk about the other stuff. Okay, so now say we wanted to rename this file. We could say just move this file, and then I don't need to specify a different path if I don't want to move it, or I can, maybe we'll just move it back to the original folder, but we'll just call this one test.bam. Now this file has moved, and if we go back, we see that file is located here and it's been renamed. So another useful feature is CP, so this is copying files. So if I wanted to make a copy of this test.bam, I could just say cp test.bam. So I specify the file that I want to copy first, and then the, excuse me, the copied file name. So I run that, look at the files, and now we see both the original and the copy there. And again, I could do a kind of chain these commands together where I copy this folder file and directly move that file to the away folder. And then if I were to go into the away, that copy is there. Yeah. Okay. So now before we start actually looking at some of these sequencing file types that you're most likely to run in, let's just kind of quickly review what sequencing is doing, because this is going to determine the steps that we need to take when we're processing the data. So when you are doing genome sequencing, RNA sequencing, any sequencing, kind of follows a similar pattern. So you take your RNA or your genomic DNA, you'd fragment it to anywhere from 50 to 200 base pair length. And then you would ligate these adapters to the ends of those fragments. And so those adapters will basically allow the reads to be, as you can see here, be amplified in the sequencing machine, and then kind of read through read by read. So it basically lets the sequencer know where to start in the read. It will read by read. And then also importantly, within these adapter tags, there's going to be a sample ID so that you can pool several samples together. And then once you're post-processing, you can sort a read that belonged to this sample here to this file, and a read that belonged to another sample to another file. So then this is going to be output to a text file. And so when you're dealing with sequencing data, you're going to not only have the nucleotide sequence of that read, but you're also going to have a quality score for each base pair there. And that just says how confident the machine is that that nucleotide is the nucleotide that it says it is. And then afterwards, we'll align this to the reference genome and be able to do any downstream quantification and analysis. So that's a very, very boiled down summary. I don't know if anyone has any questions about kind of the nuts and bolts of sequencing, we can talk about that real quick. Let's kind of pause here for a couple seconds. Okay, so let's dig into the actual file types that we'll be dealing with. So we talked about how once we have these reads, we need to find out where they belong in the genome. And that's how we, so in RNA-seq, that's how we would quantify gene expression based on the number of reads that align to that gene. And so this has already been curated, where a record will start with this caret symbol, and it'll have the identification. And then the line that follows will be the nucleotide sequence. And so a FASTA file, which is going to contain these records, it can be one record or it can be several. You could have all 23 chromosomes of the human genome in one file, and it will just identify each chromosome by a different caret with that chromosome ID. So then off the sequencer, we're going to get a FASTQ file. So this is going to be basically structured like this. So each read is going to generally be 4 lines in the file. Sometimes they can be compressed, but usually you'll see it like this. So your first line will start with the @ symbol, and this will contain the read ID, just to identify it as a unique read that was sequenced. And then we'll have the nucleotide sequence there. And then you'll have a plus, followed by the quality score for each of those nucleotides. So you can see this colon lines up with this C, this @ symbol lines up with this T. And so this scoring system is called the Fred score. And the reason this was developed is basically there's um, basically it allows for more quality metrics than there are single numbers. So basically you could have 40 plus different quality scores and they're all boiled down into a single character each, which then downstream tools know how to decode, and they can say, okay, this is maybe only a quality score of 5, but this one is a quality score of 40 or something like that. So if you want to read more about this, you can go to the Wikipedia page, kind of learn about the history of that and why that's used. But that way, if say there's some really poor quality nucleotides within the read, so maybe with this read, the first 5 nucleotides just have really bad confidence. It could basically be a 50-50 chance that it's an A or T or whatever. It will just trim these reads off and just say, hey, I can't be confident what this is, so this could mess with aligning to the genomes. I'm just going to get rid of them. And so that's the first step that you should be thinking about when you are processing sequencing data is first that initial quality control and pre-processing. So remove sequencing adapters, remove poor quality bases, because if maybe this is what aligns to the genome, but this is just kind of gibberish, this gibberish is going to prevent the rest of the read from aligning to the genome reference. Okay, so then the next file that we're going to get in this sequencing pipeline is it's called a SAM or a BAM or a CRAM file. So this is after all these reads have been aligned to the genome. We're going to get this file that's a sequence alignment map. And then a BAM file is just a binary version of this. So this condenses a maybe 10 gigabyte file down to 1 gigabyte. So it's a lot easier to move these alignment files around from maybe the supercomputer back to your personal computer. And this is where that SAM tools tool is going to come in handy. SAM tools will allow us to read a binary alignment file. A CRAM file, this is a BAM file that's compressed not as well. It's basically a bad compression that could not be as accurate as a BAM file. I would say kind of standard is to stick with BAM files. So that would be my recommendation, is not deal with a CRAM file. And so this is a command that we might use to look at this BAM file. So we can look at this test file that we had before. So I can say samtools view test.bam. And so now I can run this command and it will just spit out that whole alignment file, which could be hundreds of thousands of lines. But I want to basically send that output to some other command line commands to clean up the output so it's easier to process. And so what I'm going to use is, after that samtools command, I'm going to use this pipe symbol. So it's just the up and down symbol. It should be above your enter key. And so this is basically going to say, run this command, take the output, pass it to this next command. So this next command, because columns are separated by tabs, I'm going to kind of clean up the output so it's easier to read using this. And then finally, I want to pipe this one more time to that less command. So we're going to provide that dash capital S symbol to let us scroll up and down and sideways. I don't remember what the dash capital N is. So let's run that. And because this is a big file, this is probably a gigabyte large, it's going to take some time to read this. Alternatively, what I can do, I'll just quit out of that. And instead, I'm going to add one more pipe here. So I'm going to say head -n. So this is just going to take the first 100 lines from this output, format it, and let us read it. So that reads through a lot faster. Okay. So let's go back here. So SAM files are usually, I think it's, I don't remember the exact number, but the series of kind of standard columns and what is in each of those columns. So this first column is going to be the read ID. So if we go back up to this FASTQ file, here's that read ID for this read. And we can see that would be here to say this is the unique read. Now we have a kind of condensed numerical mapping quality number. So I think this tool is 256, I think, is a unique match that's not, that read isn't mapping to multiple locations in the genome because of maybe a repeat region or just close nucleotide identity. Then in the third column, we have the chromosome number. The fourth column is the leftmost mapping position of that read. So basically, if we look here, so we're going to skip ahead to, this is that nucleotide sequence. So this first read from this nucleotide sequence, that C, that would be at position 985. And then A would be 986. Then we have another kind of just simplified numerical score for the mapping quality. And basically a couple more columns of this metadata that I think usually you don't have to be too concerned about. Okay, so a question about how do we interpret map quality score as good or bad? Is there a threshold? So yeah, there's a couple ways. So there's kind of a gradient to scoring, and you'll be able to consider this gradient in downstream steps when you're actually quantifying these reads to a gene. So these ones you don't really have to worry about. I would say the most applicable ones to someone is in this column. So this is going to say if it's a unique match. So this is a perfect map to just one region in the genome, would be 0 or 256. Maybe 272 is, it's a perfect alignment, but it maps to 3 places in the genome. And so we don't actually know where that read originated from that we sequenced. So there's some uncertainty there. And then there's also the possibility that this is a imperfect alignment. So maybe out of the 50 nucleotides, 48 of them perfectly matched. And so downstream, you could say, I only want reads that map to a unique location and they perfectly map to that location. I only want to consider those in my downstream quantification and analysis. So some of that is also contained in this information here. So if you see there's an NM tag here. So this is number of mismatches. So with these reads, they all had 0 mismatches. So they're a perfect alignment. And all these ones that were 256 were only located at one position, where some of these ones, they had 0 mismatches, but maybe they aligned to multiple places in the genome. So any other questions about this format for the SAM and BAM files? Okay. So with a lot of, at least with sequencing analysis, a lot of the analysis that you'll want to do, this will just kind of be an intermediate step for you. It's good to know what these files are containing, but in most cases, you're just going to pass this file from your alignment software to maybe a quantification software or a sequencing coverage package to visualize maybe isoform abundance, or if you're doing genome sequencing, maybe you want to visualize a genetic deletion. So in most cases, you'll just need to pass this to the next step, but I think it's good to know what you're basically cooking with here. That's the wrong button. Okay. So then the next step that we would generally do is quantify those aligned reads to the actual transcriptome, because we want to see basically of all these reads, how many align to gene A, how many align to gene B, and then sample a sample, we can see if that gene is being upregulated or downregulated. And so we'll talk about the tool that we would use, I think, next week, but one of the important data types or file types that's already been curated that we would get from another source. So you'll see this one's being downloaded from Ensemble. So this is basically curating the human genome or yeast genome and the transcriptome annotations in one location for easy access. So this is called a GTF, or a gene transfer file. So this is going to contain all those gene records to say at this, from this coordinate to this coordinate on chromosome 1, this is gene A. And so this is where read 1 would align to in the genome, or what gene it would align to. And so you notice when I download this file, it ends in .gz. So .gz is just another compression algorithm, similar to like a zip file that you would see on your computer. And so you can just uncompress this using gzip-d. Let's just decompress the gz file. If you wanted to gzip a file back up, you would just do gzip without the -d and then package that file back up. Okay. So let's talk about, so this is how I would maybe preview what this GTF file looks like, and I could throw in that head command in between another pipe if I want to visualize it, but we can just kind of look through here on the screenshot. So you'll see here, there's a couple lines that start with that pound symbol, well, pound and exclamation mark. So these are comments. So comment lines. So these won't be, these aren't to be interpreted as an actual record, but it's just some metadata about this file. So you can see, in this case, the genome build, the genome date, and whatever other information there. So tools that we would give this to, they know how to basically ignore these metadata lines and just consider the record lines. All right, so that's, I'm gonna go back real quick. That dash capital N, I think that just lists the line numbers on the side there. Okay, so I don't exactly know why this is out of order with the chromosomes, but so in this first column, you're gonna have the chromosome number. The second column would be the annotation source. So this is the yeast genome. All these annotations were derived from SGD. Then in the third column, we have the record type. So with these, you can see here's the start and end coordinate of this record. So this gene, this is the location, start and finish. And then within each gene, so you can see these lines are for this first gene, COS7. And so here's transcript A, the location. Here's all the exons or coding space, start codon, stop codon. You'll have, in mammalian cells, you'll have UTR annotations there. I think the score column, that is more on the annotator side, not something you normally need to work or deal with. And then this column, we have the strand. Is this gene record positively or negatively stranded on the genome? Is it in frame or is it +1, +2? And then this last column is all of the metadata. So for this gene, we have the gene ID, the gene name, gene source, gene type. With transcript, we have the gene ID again, and that gene name, but we also additionally have the transcript ID and some more metadata after that. So I'll pause again real quick just in case there's any questions about this format. Okay. So then the last common file that we'd be dealing with is a delimited file. So this is just a comma separated file or tab separated file. Those commas or tabs just tells the file where's the new column, essentially. So this is basically a very basic Excel spreadsheet with all that, without all the fancy formatting. So either of these work for a lot of applications, and a tab separated file could end in .txt or .tsv. My personal preference is to always use a TSV file. And the reason for that is, say you have a gene name with a comma in the gene, maybe gene 1,6 or whatever. If you're using a CSV, halfway through that name, it might interpret that comma to say, hey, start a new column, and then everything's going to get out of alignment. Whereas with a tab separated file, so this is going to be using, instead of like maybe here's call1, comma, call2, this is going to be using this special character, this /t, call2. So you're never going to have a G name with this symbol in it. And so it's just kind of a good security net to make sure you don't read your file weird. So then usually when you're doing gene quantification, so you're quantifying gene expression, usually these will be output in some kind of text or comma or tab delimited file. Okay, so I actually demonstrated this already with viewing that BAM file using, just if you want to visualize the whole file or just visualize a chunk of that file. You could also do tail -n40 to view the last 40 records, the last 40 alignments. And so now we're going to do some, I guess, fancier searches and pipe commands from this BAM alignment file. We can determine the sex of an individual based on the number of alignments that align to the X or Y chromosome. So there are a lot of resources where you can access these already processed BAM files. So I think I'm going to recommend at the end of, for the homework section, I think it's the 1,000 Genomes Project. So you'll be able to access these anonymized human samples, and then we can do some fancy searching for the X chromosomes or Y chromosomes. So let's see. So let's take this file here. So I'm gonna visualize it, SAM tools view, provide the file name, pipe. And now I'm going to use a command line tool. So this is called grep. So this is basically your find function within command line. So this is going to say, search for any line that has a capital Y in it. And again, this is case sensitive. If I wanted to make it case insensitive, I could do -I. Or maybe I wanted to find every line that didn't have Y in the line. I could say -V. That's going to do a inverse search. But we don't need to do anything fancy for this. We're just going to do the capital Y. And then I'm going to add this formatting for visualization. And then I can run this, let this search through the file. And while this is searching, so grep can be used on any plain text file. So if you just wanted to give it a data frame that's a .tsv file, it can search through each row and find a certain string on that row. The only reason that we're able to pass this BAM file in to grep is because we're using the SAM tools view. This is going to convert that BAM file to that binary format to a human readable format and give that to grep. So it's taking a little bit longer than I expected. But while it's running, we can just look at this example output. So at the end, instead of visualizing all the reads that had Y in them, I piped that one more time to the word count function and just returned all the lines. So for the Y chromosome, we have 1,000 alignments in the sample. To the X chromosome, there are 32,000 reads. So a quick question, why do you think there are fewer reads for the Y chromosome for this individual?
The Y chromosome is small.
Yep, perfect. And just one other disclaimer. So with this, there's a good amount of reads for the Y chromosome, so we can predict that this individual is male. There are some cases where, so maybe in a female you would have 0 Y. You could also have some cases where maybe there's 20 alignments to the Y chromosome, and that just may be maybe if you had a bunch of not mismatches, but maybe you had some reads that had some mismatched nucleotides where it's not a perfect alignment. Maybe it could also be aligning somewhere else, and so it's just kind of noise in the data, or could just be weird sequencing or something like that. It could be a variety of reasons for that, but generally you should have a decent number of reads there. So here we have 24,000 reads that align to the Y chromosome in this individual. So there's that. Okay, so quickly introduce the homework. So on the supercomputer, I want you to download 3 BAM files. So you'll do this from the 1,000 Genomes project. So here's their data portal. And so I'm going to go to their final file download folder. I'll look at data. Nope, I guess the data was moved. Let's see. So basically you want to navigate through this file structure, find 3 different BAM files. And then here's one that I downloaded before. We'll go to the alignments. And here's a BAM file. You can just use these mosaic BAM files, that's fine. So I just right click on these, copy the link address, and then I can go to the supercomputer and use that download URL command again, so the curl dash capital OL, and then I'll paste that link there, and then I'll download that file for me. And then I can use the SAM tools view and so forth. And with these, so you won't run into this by just downloading 3 BAM files, but it's good practice to clear up files that you're sure you don't need anymore, because like I said before, I think you have a soft file limit of 50 gigabytes on your home directory and a hard limit of 75 gigabytes. I'll talk some more about data management, especially with sequencing data sets where you may be generating a terabyte of data for one experiment. But we'll get to that next week. So I'm going to go ahead and answer the previous question about conda with an error. And then if you have any other questions from today, let's go over those. So there's a question, conda create dash dash name class gives me an error. Conda command not found. So could you try just running conda dash dash help and see if that pops up anything? Maybe while they're testing that out, if anyone has any other questions, we can go through that.
Jordan, I had a similar problem. I couldn't get help to work, and so I did some Stack Exchange Googling and found that if I typed bash and then that moved me home or something, and then conda worked.
Okay, so I'm wondering if that is the case. It could be that conda wasn't initialized correctly, so I can just kind of show you what that was doing. So in order for conda to be able to be found anywhere and not just when I'm in the conda folder, so there's going to be a hidden file here that I can open. So I'm gonna we're not going to get into the specifics here, but I'm going to use this. I guess we can just use cat. So cat dot bash underscore profile. Enter. I guess I should have used the less command. I can just pipe it to less here. Where is it? I guess it would be found, not bash profile, but bash RC. So if you open up bash RC and go to the bottom, you should have this conda initialize section in the file. And so this is when you log into the supercomputer, it's going to say, here's where conda is, here's where that conda.sh command is. So instead of having to run bash conda.sh, you can just run conda as a shortcut.
Okay. Even when I was in the folder where I guess installed mini conda, I couldn't run, the conda command wasn't working.
Okay, did you have to go into the mini conda folder to run it?
Even when I did that, it didn't work. But after I did the bash thing, it did work.
Okay, interesting. I am not totally sure. I mean, if you want to either copy kind of some of your output to the, okay, so someone's getting the same error. That is very interesting. Okay. So there's a couple options. If we want to, you can share your screen, just share your terminal window, and we can kind of troubleshoot this step by step. Or if you just want to copy and paste what you tried in the Slack channel, we can go through it there. Whatever you're comfortable with, we can do that. Or if you also, if you want to just personal message me on Slack, your UID, I can look into your, can you just give the URL for installing Conda for Windows? I think I missed it when you were talking about it. Oh, okay. Yeah, I can give that URL again for downloading Conda.
I think, so it is working now with this post having typed bash. So maybe when I reopen this to do my homework, I will see if I have to do that again. And if so, I can share with the group what's going on.
Okay, I think I know what's going on. So if you log out and log back in, it
Yeah,
may...
I tried that too.
Okay, and that didn't work still?
I tried that before the bash trick, and that did not work. So I kept reading the next comments on Stack Exchange, and this one worked.
Okay. Yeah, maybe let's, after this session, we can work through that and figure out why you can't just use conda, the command. And then once we figure that out, we can just post the solution on the Slack channel in case anyone else runs into that. So another question, give the URL for installing conda for Windows. So if you're on a Windows computer, but you're installing conda on the supercomputer, you need to use the Linux file. But let's see if I can add that again. So I will copy this URL to the Slack channel. I'm going to put it in there's a command line channel specifically, just so we can kind of keep these commands organized. Okay, so that's available in the Slack channel in case people want to use that. Okay. Any other questions about file types or tools or any other issues that you ran into if you're following along? Okay, if you have any more questions, if you're running into issues, feel free to, you can just post them on that command line channel. And maybe if someone else has a solution, they can post that there. Or if people are getting stuck, I can jump in with this, getting the conda program set up. I'll troubleshoot that immediately with people just so you're not stuck on that step. But if there's no other questions, we can finish for the day. And I will see you on Friday, either at 10 a.m. or 5 p.m., depending on which session you decide to come to. And we can just do some more troubleshooting and review, kind of walk step by step through some of the homework problems or any other test cases you want to play around with. Okay, cool. Well, I'll see y'all later.